Hello, everyone. This is Frank Ambrosio, bioinformatics scientist with Theogen Genomics, and welcome to the first session of our two-part bioinformatics fundamentals course. Starting things off, I am Frank Ambrosio. I am a bioinformatics scientist with Theogen Genomics, and this course is going to focus on pathogen genomics specifically. So we're going to use bioinformatics to look at clinical data, public health data, and learn how to investigate outbreaks and perform surveillance activities. So the main course objective here is to learn the fundamentals of bioinformatics and next-generation sequencing and how we can apply those concepts in the field of pathogen genomics. Like I said earlier, this is a two-part series. So the first part is going to be the fundamentals of pathogen genomics. We will cover molecular biology, next-generation sequencing, pathogen genomics in general, and then finish things up with the fundamental concepts of bioinformatics. During the second section, we will go over genomic epidemiology. And that's usually downstream of bioinformatic genomic characterization. So this is a logical flow for this content to be delivered. So we'll start with genomic epidemiology in week 2. We'll go over phylogenetic tree construction, which is one of the primary tools used by genomic epidemiologists in both outbreak investigations and surveillance activities. And then we will cover specifically some case studies on outbreak investigations and genomic surveillance. Throughout both of these sessions, we have a list of resources that will definitely be useful if this is your first time being exposed to bioinformatics content. So first up, we have the Staff B Glossary of Bioinformatics Terms. That's the State Public Health Bioinformatics Group. They have produced a glossary of bioinformatics terms that will be helpful throughout learning bioinformatics. There's a ton of terminology that's very specific to the field, so definitely check that out if you're running into any terminology issues. Next up, we have the Theogen Public Health Resources. Full disclosure, I work for Theogen, but this is a fantastic resource to learn about bioinformatics pipelines. We have great documentation for all of our workflows. So if you are considering starting to build your own workflow or you're interested in how these workflows work under the hood, our documentation site is a great place for that. And then finally, the Public Health Alliance for Genomic Epidemiology has a group called the Bioinformatics Pipelines and Visualization Group. And this is a great place to go if you're looking for a set of standards or a roadmap for how to use bioinformatics and genomic epidemiology to perform these outbreak investigations and surveillance activities. So kicking things off with our bioinformatics fundamentals, let's start with molecular biology. So here we have the most fundamental of all fundamentals, the central dogma of life. So when it comes to molecular biology, this is really where everything starts. All pathogens have genomic material. DNA is typically eukaryotic genomic material. DNA is transcribed into RNA and then finally translated into proteins. That concept is the central dogma of life. So just to make sure we're all on the same page here, DNA is the primary source of genomic material, RNA is the transcription, but RNA can also be the primary source for something like a virus, famously SARS-CoV-2 as an RNA genome. Finally, the functional pieces of biology here, the proteins perform all of the cellular functions required to sustain life. So here is a visualization of that exact concept. We start with DNA, we transcribe to RNA, and then we translate the RNA to protein. For a virus that has an RNA genome, this step can often be skipped. The transcription step can often be skipped, and RNA can be directly translated to protein. Several viruses actually need to create DNA first so that it can be transcribed and translated into protein. So this process of transcription and reverse transcription happens all the time in biology. So that should be kept in mind as we go through this entire course. Also, I want to make sure that when we're doing these DNA sequencing, RNA sequencing, or even protein detection assays, we understand what we're detecting. So when we detect DNA, when we sequence DNA and we analyze it, we learn something about the proteins that our pathogen is capable of making. So if there's a protein that is capable of digesting an antibiotic, if we see the code for that protein in the DNA, we know that that pathogen has the phenotype of antimicrobial resistance because it's producing a protein that will digest the antimicrobial when it sees it. So that's just an example. There's many things we can learn about pathogens from their DNA, and that is primarily because the functional units of the cell, the proteins, are encoded in genes in the DNA. So how do we use this molecular biology? So we can detect outbreaks very early using molecular biology, and we can be very accurate with that detection. If someone comes in coughing and sneezing, there's a wide variety of pathogens that could be causing that, but by using molecular biology, we're able to pinpoint the causative agent of the infection. We can also monitor diseases spreading over time. So if you want to get a better idea of how a pathogen propagates through a population, we can sample the population and we can sequence the DNA from those samples, and we can learn, first of all, about how the disease is propagating through the population, but also how it's evolving over time, gaining those little mutations, single nucleotide polymorphism changes in the genome that may confer new phenotypic characteristics. And then finally, that brings us to pathogen characterization. This is where we want to learn about those phenotypic characteristics. Again, the example I gave earlier, if a pathogen contains a gene that produces an antimicrobial resistance protein, so a protein that digests an antimicrobial or breaks down an antimicrobial in some way, being able to detect that in the genome will inform us not to use the antimicrobial that is susceptible to that degradation or digestion by the pathogen enzyme. So molecular biology does have some limitations. First of all, of course, the infrastructure requirements. You're going to need a laboratory to do molecular biology, and not everyone has a laboratory. You also need very well-trained personnel. This is not the type of activity that you can just pick up a pipette and start squirting reagents into tubes. Typically, it requires several years of specialized training to produce a competent laboratorian performing molecular biology. And that is a result of the complexity and expertise required. The assays are often long and complex. Sometimes they take up to a week to perform all of the necessary steps to get to fully sequenced genomic material. So you'll start off with either a bacterial isolate or a plate, or even just a specimen itself directly from the patient. And from there, you need to extract the genomic material. You might need to grow up the bacteria on a blood auger plate or in a tube, and then extract the genomic material, isolate the genomic material from all the other gunk in the tube. And then you need to prepare the genomic material for sequencing. So all of that can be very complex, and even launching the sequencer can be a bit troublesome at times. So it's a very complex procedure, and that really limits the number of molecular biologists we have out there. And then finally, the cost. Every time you run a sequencer, it's going to cost hundreds, if not thousands of dollars. So it's not the most accessible assay to every laboratory around the world. And that is being addressed with some newer technologies. So we'll talk about that in a bit. So to understand the enormous advantage conferred by having access to next generation sequencing, it's good to look back at the traditional laboratory assays that were used prior to next generation sequencing's invention. So pulse field gel electrophoresis, PFGE. Many of you will have heard of this before. What it does is it separates DNA fragments by size. By putting the DNA in a gel block and running an electric current across that gel block, you get the DNA to start moving across. The larger fragments will move slower than the smaller fragments. And in that way, you can get a banding pattern where the smaller fragments are bands further down the gel block, the larger fragments are less far down the block. And you get a pattern, you get a recognizable pattern based on the sizes of the DNA fragments. Now, if you see this fragmentation pattern multiple times in multiple different pathogens, after breaking up the DNA, using an enzyme that fragments the DNA at a very specific site, you can get a good idea of what the DNA or similarities between the genomes that have been fragmented and spread out using this gel electrophoresis. So these banding patterns are often referred to as genetic fingerprints. There are databases of these banding patterns that are depending on which genome you've used and which enzyme to break up the genome into fragments you've used. With that database of known genetic fingerprints, we can fragment a genome using an enzyme that breaks it up at specific points. Specific motifs throughout the genome will be recognized by that enzyme. It will break it up into specific fragments. We load that into a gel block. We produce the banding pattern, and we compare it to the database of known genetic fingerprints. And in that way, we can identify pathogens. And this was a fantastic technique used for decades. It was really good at identifying clusters of similarly related pathogens. However, we really didn't get the resolution that you can get from sequencing the entire genome. And it did require these databases of known banding patterns. So if you saw a banding pattern that was not in the database, you really couldn't do much with it besides add it to the database. So this is what these banding patterns look like. These top wells up here are where you would put your fragmented genomes. So again, you would take a genome from a pathogen, add some enzyme to the tomb that would chop up the genome at very specific sequence locations. And then you would run an electric current across this gel block with the DNA in one of these top wells. These black rectangles here are actually indentations in the gel block. And then the electric current would run across top to bottom here, spreading out the fragments of genetic material until you get this unique banding pattern. You can see matching banding patterns between many of these. This middle one is the control here. You can tell it's the control because of the almost evenly distributed bands. And what the control is, you have known fragment sizes in the control. So when you see that banding pattern, you know anything near this band or near this band in the control is roughly the same size based on the amount of movement it's had through the gel block. So from there, we compare this banding pattern to our database of known genetic fingerprints, and we can determine what pathogen is here. And this, again, was very useful for a long time, just lacked the resolution that we need in order to fully characterize our pathogens. Next up, we had PCR, polymerase chain reaction, one of the craziest reactions that we've ever discovered as humans. What polymerase chain reaction does is it amplifies DNA. It's essentially the process of making many copies of DNA, and it happens at an exponential rate. So what we can do is from a very, very small amount of input genomic material, we can detect whether or not a pathogen is there. A very specific genomic sequence is there. So if we have a very specific genomic sequence we know is unique to Klebsiella or another pathogen, we can use PCR to detect if it is in our sample. So that's very good for identifying pathogens and detecting pathogens in low quantity. And we have seen PCR get used throughout the pandemic. PCR tests were available for SARS-CoV-2. So this is still in use today. It is, I would say, one of the industry standards for pathogen detection in both clinical settings and public health settings. So this is a fantastic assay. It can be used to identify specific genetic markers as well. So not just detecting a pathogen, but detecting a specific gene or a specific sequence of DNA in the pathogen's genome, like resistance genes. I'll keep coming back to that example throughout these sessions. One of the big advantages of PCR is it is a fairly simple assay to run. And I don't mean to disparage all of the laboratory scientists who have struggled with PCR. I am one of them. But compared to next generation sequencing, it requires less laboratory hands-on time than a next generation sequencing prep. And that means that you can rapidly screen large numbers of samples for very specific genetic markers, making it an amazing molecular biology tool. Now, PCR amplicons, so the region that gets amplified, that is limited. You have a hard time amplifying a full bacterial genome. I know there are a few groups working on this out there in academia right now, but as it stands, it is difficult to amplify an entire bacterial genome using amplicons. So PCR assays typically target a small region under 2,000 base pairs. So that is really limiting when your average bacterial genome is between 4 and 5 million base pairs. So in order to get that greater level of resolution and more comprehensive picture of the genomic material of our pathogen, next generation sequencing is the tool of choice. So that brings us to next generation sequencing. First thing next generation sequencing was not the first sequencing technology. There were several sequencing technologies that came before it. However, it was a massive jump in both accuracy and the amount of sequencing you could get per dollar. So in terms of cost and accuracy, next generation sequencing was this massive jump from previous sequencing technologies. And it made it available to laboratories around the world. So by providing accurate genomic sequence information, we could reliably characterize pathogens based on their genomes. It is a faster methodology than many of the legacy sequencing technologies. So that was another huge advantage, is you could sequence many samples on a single sequencer. Having the full genome sequence reveals genetic variations that can confer all sorts of phenotypic traits. Again, the example we keep using here is drug resistance, but there are many other phenotypic traits which can be conferred, such as increased virulence, transmissibility, and resistance to environmental factors. So comparing these genome sequences allows us to determine the single nucleotide polymorphism differences between them. And by doing that, we can identify closely related samples. And in theory, we can reconstruct transmission pathways, transmission networks, based entirely on the genomic sequencing data. Now, when you take that genomic sequence transmission pathway and you combine it with traditional epidemiological data, you get a much more robust idea of how a pathogen is spreading through your community. And this can be within a country, or it can be within a single facility. Having that genomic data really allows fine resolution transmission network tracking. So traditional methodologies have done amazing work for the field of public health and clinical microbiology. The limited pathogen scope is an issue, and the lack of complete genetic information is an issue, but these tools are still in use today, and they still do a lot for the microbiology community. Next generation sequencing, however, has given us a whole new skill set, a whole new set of abilities. So the broad pathogen identification is an obvious, enormous advantage. In order to use PCR or PFGE, you have to know something about the pathogen a priori. For example, with PFGE, you need to know the banding pattern that will result from the fragmented genomic material passing through the gel block prior to being able to say anything about the pathogen that you've just run through the gel electrophoresis machine. If there's no banding pattern that matches your banding pattern in the database, then you can add yours to the database, but you really don't learn anything about the pathogen itself. Same thing with PCR. You need to have primers that are capable of amplifying a very specific genomic sequence. If you don't know the pathogen's genome in advance, you really can't create those amplicons, those primers to amplify those amplicons, and having a PCR assay is going to be very difficult. But with next generation sequencing, you don't need to know anything about the pathogen a priori. You can immediately go into sequencing. There are certain aspects of the pathogen that you will need to understand, like is it gram positive or gram negative? That will inform your extraction methodology and your library prep, and that will impact how much genomic material you yield out of your sequencing run. So that being said, next generation sequencing allows a much more pathogen agnostic analysis. So many pathogens are difficult to extract and difficult to work with in the lab. Some grow faster, some grow slower, but they all have genomic material. Next generation sequencing is a good methodology for analyzing all of them. The faster results, that is debatable. Depends on which library preparation technique you're using, but you will get more results. You will get much more comprehensive results. So you will analyze the entire genome. You'll learn things about the pathogen that don't require knowing things about the pathogen in advance. So if there are genes that are homologous to antimicrobial resistance genes in our databases, it doesn't need to be from the same pathogen. If it has the same AMR gene, we can typically identify it with a high degree of accuracy. So that's a nice segue into next generation sequencing. I am very passionate about next generation sequencing. I think it's one of the greatest inventions of our time. So a little bit of history about next generation sequencing. This is the fun slide that I added for myself. So first nucleotide sequence ever recorded was in 1973, Walter Gilbert, kudos Walter. It was a 24 base pair sequence of the DNA LAC operator, which I believe is some sort of lactase involved operator. And 24 base pairs is not bad. That's not bad for 1973. However, a few years later, Fred Sanger, he sequenced the first bacteriophage complete genome, 5.4 thousand base pairs. So in just 4 years, we went from 24 base pairs to 5.4 thousand. And then in 1995, we have the first complete bacterial sequence, Haemophilus influenza, at 1.8 million base pairs. So we can see there is a fairly rapid increase from 24 base pairs up to 1.8 million. This sequencing technology, everyone recognized the value of it early on. And then we saw massive jumps in this technology starting in 2000. So in 2000, next generation sequencing emerges. Everyone knows the story of Illumina. If you don't, I highly recommend looking it up. It's a very interesting story. In 2008, James Watson of Watson and Crick and Franklin fame was handed his own genome on a hard drive. And then in 2008, 2009, that's when we saw the third generation, so long read sequencers emerging. And I wouldn't say that third generation is separate from next generation, even though it sounds like that. In my mind, any sequencing technology that can accurately sequence genomic material is part of that next generation. So this is the obligatory cost of sequencing curve that has to be in every bioinformatics fundamentals course. So in 2000 or so, we saw next generation sequencing emerge, and it cost $100 million to sequence a human genome. And it was about that same scale of cost. We saw it drop down to about 10 million in 2007, 2008. That's when James Watson was handed his genome on a hard drive. But then the MiSeq emerged, and essentially that's what you're seeing here with this clip. Illumina released their next generation sequencing platform, and we saw the cost of sequencing a human genome plummet essentially overnight. So we have violated Moore's law for the past decade or so at this point, and that is in large part due to the emergence of Illumina and these third generation long read sequencers. So how does next generation sequencing work? This is meant to be a course where we learn about these tools, we learn how these tools work so that we can minimally interpret analyses, analytical outputs from these types of assays. So how does next generation sequencing work? There are several phases. The first of which is DNA RNA extraction. It's debatable that upstream of this, you can include the isolating of bacteria or the growing up of bacteria or viruses in the laboratory as part of this. However, today I'm going to start from extraction. So starting with extraction, there are many different extraction methods. There are mechanical extraction and chemical extraction methods typically used. Once you've extracted the genomic material, you want to isolate it and prepare it for sequencing. Isolating it and preparing it for sequencing all fall under the category of library preparation. And then once you've prepared the genomic material for sequencing, you can put it on the sequencing device, run the sequencing assay, and then you're ready for your analysis. So out of the sequencer, you get your genomic sequences in the form of reads. Now, part of the library preparation process is breaking up the genome into smaller chunks for sequencing. We then sequence those smaller chunks, and that is what we get out. They are called reads when they come out of the sequencer. Those reads then need to be reconstructed into the pathogen genomes in order to perform analyses with them. They don't need to be. You can perform assembly-free analyses, but typically the process involves assembling those short read fragments into a larger genome. It's also worth mentioning at this point that the genome is sequenced more than once. During the library preparation process, we amplify the genome so that we have many copies of it, and then we fragment them all up and sequence everything. So if you imagine taking a bunch of sheets of paper and shredding all of them, and say they all have the exact same message on it, shredding them 100 copies of the same piece of paper, and then trying to put them back together into one functional copy of that piece of paper. That's essentially what we're trying to do with the assembly process. There are many other steps in bioinformatic analyses, many other analytical modules that we can discuss, and we'll get to that in the latter section of today's lecture. And then finally, once we have our genomic characterization, we have our fully assembled genomes, we go into genomic epidemiology, which is the process of using those genomes as a proxy for potential transmission events. So any two genomes, the closer they are genomically, the more likely that there was a transmission event between the two individuals from which the samples were originally taken. And by using genomic distance as a proxy for likelihood of a transmission event, we can get really high resolution transmission networks through a population. Depending on the sampling frequency of that population, we can construct these transmission networks and oftentimes pinpoint the exact introduction event into a population. So starting with the DNA or RNA extraction, we need to obtain the DNA from the biological sample. So we all know that in a bacteria or a virus, the genomic material is inside of a envelope or a cell wall, and that needs to be broken down without destroying the genomic material. So this process, there are many ways to do it. The most crude of which is called bead beading. You literally put small beads in a tube with the biological sample, put it on a shaker machine that vibrates the tube, and it smashes apart all of the bacteria, and then you run it through a filter that will extract just the genomic material and wash away everything else. A more elegant solution, however, much more labor intensive in the laboratory, is something like a phenylchloroform extraction. That involves many, many pipetting steps, but essentially what you're doing is dissolving that outer layer of the bacteria so that you can get at the genetic material without destroying. Next, we have library preparation. So once we have the genomic material out of the bacteria, part of what we want to do is fragment it into smaller segments, and this can be done in a couple of different ways. The first of which is enzymatic. You can have the same enzymes that were used in PFGE to fragment the genome at very specific locations can be used to fragment the DNA for sequencing. I know that there is a process in one of the Illumina kits called tagmentation, where the DNA is fragmented and tagged at the same time. And when I say tagged, I mean tagged with these adapters and or barcodes. I believe the tagmentation applies the adapter only. But the adapter is going to be used to connect the DNA fragment to the flow cell in the sequencer where the sequencing will actually take place. So we prepare the genomic material for the sequencing run by fragmenting it into smaller segments, either using that enzyme, or there's another process that we can use called sonication. And that's where we put the genomic material in a tube, we put the tube in some water, and we shoot sound waves through the water to break up the DNA into specified lengths. And it was always fascinating to me, by using the resonant frequencies and the sound waves, we could select the length of genomic material that we would like in our fragments once they're broken up. From there, we amplify we attach the adapters and barcodes. The adapters are, again, to allow those fragments to attach to the flow cell in the sequencer. The barcode is so that we can differentiate between different samples in the same sequencing run. So you can sequence multiple different pathogens on the same flow cell, but by attaching a genetic barcode, essentially, to these fragments, we're able to differentiate where the fragments first came from, the source sample for the fragments. From there, we perform amplification. So you have your genomic fragment from your pathogen genome with the adapter and the barcode on the ends, and we amplify that using PCR. Then we have a lot of copies of all of those fragments, and we load them all onto the sequencer, and we sequence as much genomic material from our pathogens as we can, given the amount of resources in our sequencing platform. So sequencing by synthesis, the Illumina process is a really cool technological feat. So is nanopore sequencing. I strongly encourage, there are videos online that describe these things in great detail. There's a great video about sequencing by synthesis on the Illumina website, actually. If you want to see bridge amplification and all of the steps that occur on the flow cell of the sequencer, I strongly encourage checking that video out. So to start the sequencing run, we pool our libraries. That's what it's called after you've performed that library prep. Once you have your adapters and barcodes attached to those fragments and you have amplified those fragments up, you have enough to go into sequencing. The amount of genomic material that you load onto the sequencer will influence the amount of reads, the amount of genomic material that you get out. So reads in equals gigabytes out. If we have multiple samples going onto a single sequencing run, we have to pool the libraries from each of these samples together. We denature the libraries, and then we load them all onto the sequencer. So once they're loaded onto the sequencer, sequencing by synthesis or nanopore sequencing, those are the two that I am most familiar with. There are other sequencing methodologies, but for now, let's just focus on Illumina and nanopore. This sequencing will take place, and read files, also called FASTQ files, are generated. So sequencing inside of the sequencer has many steps. Let's just go over some of the main steps here. So first up, we have cluster generation. And this is a very interesting step here. Cluster generation starts, or apologies, cluster generation starts with the fragments binding to the flow cell and then undergoing this process of bridge amplification. Essentially what happens is the fragment uses the adapter to bind to the flow cell, folds over itself, and connects again to the flow cell, creates a copy of itself. Then they both come up and they go through that same process again and again. Because it starts from one spot and spreads out with that bridge amplification, you get clusters on the flow cell. And this is happening at the molecular scale. So you have these tiny little dots of exact copies of the fragments that you have prepared in your libraries all over these flow cells. Then the next step is called sequencing by synthesis. And this is specifically for an Illumina platform, I should mention. Sequencing by synthesis. So once you have these clusters, what takes place there, each of those fragments within the cluster is then copied. Fluorescently labeled nucleotides will be added or bound to the copies, to the fragments that have created those clusters. And they will emit light signals that are detectable by the optical device within the sequencer. The image acquisition step is the step that occurs every round of sequencing by synthesis. So essentially what happens is you have a cluster of fragments. Each fragment has a nucleotide bind to it, it fluoresces, and a picture is taken. And based on the color of the fluorescence, the device can tell which nucleotide was added. And by telling which nucleotide was added, because nucleotides are complementary, we know what nucleotide was in the fragment. And once we add all of the nucleotides along the entire fragment, we take a picture at each step, we can then deduce what the entire sequence of the fragment was. And we're doing this for hundreds of thousands of clusters all over a flow cell at the same time. So it's a very high-powered optical device inside of the sequencer. It's very good at differentiating between different colors, and multiple different clusters will be fluorescing with different colors at the same time, because not all of those fragments have the same sequence. Now, if you have a pathogen that is very clonal, and you're sequencing 8 different copies of it, or 8 different samples, and each of those genomes is going to be very similar, you are going to want to add some noise to that signal so that 2 clusters that are emitting the same exact color don't wash each other out. And you can differentiate between the clusters a little bit. So oftentimes we'll add in an alternative genome, a bit of genomic material that is not any of our samples. Something like Phi-X, which is a phage genome, that just creates some other colors on that flow cell so that we can get a better image. The image acquisition phase occurs in between each sequencing by synthesis phase. So we add a nucleotide, it fluoresces, we take a picture, that picture is saved, once we have all of the pictures of each nucleotide being added along our fragment, typically about 250 or so along our fragment, then we use decoding software to convert those images into sequence data. The sequence data is then generated in the form of FASTQ files. So if you see a file ending that is .fastq or .fastqgz, that is usually raw sequencing data. In addition, based on the clarity of the images, we include in those FASTQ files a set of quality scores. And those quality scores can be used in downstream analyses to filter out reads that the images were not very clear. And in that way, we can only use the best quality data going into our downstream analysis. So what is a FASTQ file? FASTQ file, one of my favorite file types. Here at the top, you can see we have an identifier for each read. And I should mention, FASTQ files are the standard format for storing raw sequencing data. So these are usually the inputs to a bioinformatics workflow. At the top line of a FASTQ file, you will have a sequence identifier. You have a sequence identifier for each read stored within a FASTQ file. FASTQ files are usually sample specific. So you'll have sample 1, 2, 3. Sample 1, the FASTQ file will have all of the reads from sample 1. Now, I should mention with Illumina sequencing, it is typically paired end sequencing. I can go over this a bit more in detail, but suffice it to say for now, these are reverse sections of the genome that are separated by a spacer, and they are used together in all bioinformatic analyses. So for each sample, you will have a pair of FASTQ files. Illumina sequencing is often called paired end sequencing. You can perform single end sequencing with an Illumina device, but the paired end sequencing nature, again, I strongly encourage you to watch that sequencing by synthesis video on the Illumina website. You will understand it much better with the visualization. Now, once you have the pair of FASTQ files for each sample, if you want to know what's inside the file, at the top line, it will be an identifier for each read, including some information about the length and where the read came from. Then you will have the actual sequence as determined by the images in the sequencing device, a plus sign just to separate, and then your quality scores encoded using ASCII encoding. Each of these characters has a numerical value associated with it. That numerical value is your quality score, which is often called a Q score in the world of sequencing. That Q score is on a log scale. So a Q30 is the typical standard threshold for good quality reads. A Q30 equates to 99.9% accuracy. A Q20 is 99.0% accuracy. A Q10 is 90.0% accuracy. So the likelihood of having the correct base call given a Q30 is going to be 99.9. Now, if we're sequencing the genome multiple times at every location, we could take 99.9% accuracy and increase that dramatically with each layer that we add to that sequencing. So as you can see here, we have these 4 lines repeated multiple times throughout the FASTQ file with different sequences and quality scores, of course, but this is the structure of a FASTQ file. Now, the reason I have this here is just to avoid confusion down the road. A FASTA file is typically the file type where fully assembled genomes are stored. So you have your raw sequencing reads in a FASTQ file, but in a FASTA file, we have the assembled genomes. Now, a FASTA file is structured differently from a FASTQ file. There are not typically quality scores in a FASTA file. You simply have your header, which is, again, some identification information about what you're seeing here, and you have the sequence. And what you'll notice is we have multiple sequences in here. That is not to say we have multiple assemblies from multiple different pathogens. What it is when you assemble a genome, you will not usually get a fully circularized, complete bacterial genome. What you will get is a set of what are called contigs, and contigs are contiguous sequences in your assembly. So we sequence a ton of genomic material from an individual sample. We start assembling that genomic material, and often we won't get the whole genome in one straight contiguous chunk. But what we do is we take each of those contiguous chunks, we sort them by length. We put the longest one at the top of the FASTA file, and that is your assembly. So it may not be the entire genome. There might be gaps, there might be breakpoints. What you have here is all of the assembled genomic data. So again, in the FASTA file, you have some descriptive information in the header and then the sequence. So that brings us to the final phase of next generation sequencing, bioinformatic analysis. That process of assembling a genome from reads is it's a complex process. There are many different computational approaches to doing that, but we'll go over some of them today. So fundamental concepts of bioinformatics. Again, I consider myself a bioinformatics scientist. Bioinformatics science is the study of biological material, DNA, RNA, proteins, and using informatics to analyze these otherwise unfathomable amounts of data. So bioinformatics analysis, starting with data pre-processing, we want to remove any low quality reads, adapters, and trim the sequences based on quality. So the old adage, bad data in, bad data out, we want to remove any of our low quality reads. We want to trim off the adapters because they're not part of the original biological genome, and we want to trim any low quality ends of the sequencing. Often when we perform sequencing by synthesis, the ends of the reads can be lower quality than the middle of the read, so we don't want to throw the whole thing out. We trim off the ends. Next up, we have assembly. So if you're using de novo assembly, that is the process of assembling a genome without a reference genome. If you're using reference guided assembly, essentially what you do is you take a complete genome that matches the species of the pathogen or even the subtype of the pathogen in question, and you align your reads to that reference guided genome. So that reference genome, that's called reference guided assembly. De novo assembly is the process of using the overlapping bits of the reads to string together those contigs that we discussed a few minutes ago. Those contigs can then be cleaned up by additional sequencing runs and circularized into a complete bacterial genome or completed if a viral or human genome. From there, there are many different ways that you can take bioinformatic analysis. Typically, the next step is taxonomic identification, where you want to know the genus and species of the sample. You want to make sure that your pathogen is what you think it is, and if it's not, you want to know what it is. Then we go through the genome and we annotate genes and genetic variants. We can predict the function of certain genes and certain gene mutations. And then we go into the process of genomic characterization. So genomic characterization is taking all of those variants, all of those genes that we found, and assigning phenotypic qualities to them, and making some prediction about how this pathogen will behave under certain conditions, whether being exposed to antibiotics or other factors. So data pre-processing. What does it look like? We have to do adapter and quality trimming. Again, the adapters come from our sequencing kit, our library prep kit, and we want to trim off any low quality sections of the genome, any low quality sections of the reads. So we remove all of the adapters, we remove reads that the whole thing was too low quality, we remove sections of the reads that were too low quality, and we trim off those known adapter sequences. From there, we want to look at the quality. We want to see, okay, we've trimmed off the low quality, but now what's the average quality of all of our reads in this sequencing assay? And again, Q30 is the threshold of choice in bioinformatics. Here you can see it is on a log scale. I don't think I've ever seen a sequencing run at Q60, but that would be amazing. Typically, with Illumina sequencing, you'll see Q scores between 30 and 35. With nanopore sequencing, it used to be between Q10 and Q15. However, with the recent release of their newest chemistry and flow cell, we have seen an increase up to around Q20. So the next thing to take into consideration is the amount of genomic sequencing data that you've produced in your sequencing run. So we have a genome that is 100 nucleotides long. What we'll do is we'll sequence it at 10x coverage. So we'll sequence 1,000 nucleotides to get at a sequence that's 100 nucleotides long. So we have the total number of reads in our sequencing run times the length of that read. That gives us the total nucleotide sequenced. We take the total nucleotide sequenced and we divide by the genome size, and that gives you your estimated genome coverage. So Our total number of reads, let's say we have 10 reads and they're each 10 base pairs long. That gives us 100 nucleotide sequence. Let's say we have 10 reads and they're 100 nucleotides long. That gives us 1,000 nucleotide sequence. Take 1,000 nucleotide sequenced and we take our estimated genome size of 100 and we divide them. What we have is an estimated genome coverage of 10x. So to go through that again, we have 10 reads, they're 100 nucleotides long, gives us 1,000 nucleotide sequence total. Our original biological genome is 100 nucleotides long, which means we have sequenced that genome 10 times in our sequencing. That's important to understand because the depth of coverage of your sequencing run is a factor in how confident you can be in the sequencing and downstream results. So then we go into de novo assembly, reconstructing the sequence from all of these short read fragments. There are many different algorithms for doing this. So to just go over the general process that all of the algorithms attempt to do, you have your extracted bacterial genome. Typically it's a bit fragmented, but hopefully you can get a few complete bacterial genomes in your extraction. From there, you prepare your libraries, which involves fragmenting the DNA to a very specific length. Then you perform your sequencing run on your libraries and you get your sequencing reads, which are corresponding to their biological counterparts, the DNA fragments from your genome. And what we want to do is take that circle all the way around to a complete genome assembly. We have our biological and our digital counterparts here, if you will. The most common approach, so assembly occurs at this third arrow. The most common approach to assembly is to use what's called a De Bruijn graph or a De Bruijn graph. There's much debate on how that word is actually pronounced, but it is the process of using the overlapping ends of the sequencing reads to string together these contiguous sections of genomes. The math there is a bit out there. If anyone is like really into math, you can definitely look up De Bruijn graph assembly and you will have a field day. There's tons of materials out there on it. So FASTA files. The FASTA file contains these contigs that have been constructed by your assembly. What we can do with a FASTA file is perform analysis, identification of genes. We can compare FASTA files together to identify polymorphisms. So I just wanted to bring us back to the FASTA file because at this point we are looking at assembly. Remember, we started with FASTQ files and we have constructed them into a set of contigs or contiguous sequence sections, and we have added them to our FASTA file. So assembly also receives some quality control. Remember, we removed any low quality sections of our reads, but the assemblies can also have different levels of quality. So remember, I mentioned the depth of coverage is super important. So the number of times you have sequenced the entire genome is another way of thinking about this. The average depth of coverage at each loci in the genome. It should be stated that this is not consistent across the entire genome. You will have regions of high coverage and regions of low coverage. So regions of your genome that have been sequenced more than other sections. So you can be more confident in the ultimate base calls and the more the areas of the genome that have been sequenced with greater depth. The number of contigs is going to be a great measure of how well you did at sequencing the whole genome. If you have one contig, congratulations, you have done perfect. You have sequenced the entire bacterial genome in completeness. That is typically not the case. Usually you'll see anywhere from 20 to 500 contigs in the resulting assembly. This is highly dependent on the quality of your sequencing run, how much sequencing data you were able to produce, and on the how much sequencing data you were able to produce, and it's also dependent on the sequencing technology that you're using. So the length of the reads. Illumina reads are typically between 150 and 350 base pairs in length. Nanopore reads can get up to 3 million base pairs. I'm not sure what the record is right now for the longest nanopore read, but it is up in the millions. Average read quality, so the Q-score of all the bases in your FASTQ files will, of course, affect your downstream assembly. And then the assembly length, it is more difficult to assemble longer genomes than it is to assemble shorter genomes. That should be understood. We have some measures of quality control. Every time we sequence a genome, we want to know that we have the genome represented we want to make sure that we have the whole genome represented in the sequence data. And to assess that, we look at the genome length and the depth of coverage across the genome. We want to know how fragmented, how contiguous is the genome. And to look at that, we look at the number of contigs. Is the assembly accurate on a per base basis? And to get at that, we look at average read quality going in and our ability to taxonomically identify based on the genome. And then one thing we should always take into consideration, has there been contamination? Do we have enough reads from our target organism? I guess I haven't mentioned this yet, but in the event that you contaminate your sample or there's an environmental contamination, you will be sequencing additional pathogens in that sequencing. We remove those non-target reads bioinformatically, but often after removing those reads, you are left with not enough reads to proceed with an actual bioinformatic analysis. So it's important to assess, do we have enough reads from the target organism to proceed with our analysis? And to do that, we look at the final assembly length and the ability to taxonomically identify once again. So taxonomic identification, how does this occur? In our Theogen workflows, we use a tool called Gambit and a tool called average nucleotide identity, or a technique, I should say, called average nucleotide identity. Gambit is a super fast k-mer-based taxon identification tool. If you're unfamiliar with k-mers, all it is a stretch of DNA of length k. So a 10-mer is 10 nucleotides. What it does, it has a database of these k-mers that it compares against the k-mers from your sample, and it can accurately identify at the species level. Average nucleotide identity is a more computationally intensive process that does a full-length comparison of your genome against a database of reference genomes. So taxonomic assignment with ANI involves producing a sequencing assembly and having a reference database of known genomes. So we have our E. coli reference, our Listeria reference, our Salmonella reference, and our query sequence. We compare our input sequence against each of the sequences in our database by aligning them, and we get a percent match. And based on the highest percent match, we know what our sample is. ANI is considered the gold standard for in silico taxonomic assignment. It is used in a wide variety of bioinformatics workflows. It is relatively compute intensive. Smaller curated data sets are typically recommended if you're just running ANI for quality control. So for Gambit, our sample is a compressed representation here. Essentially what this compressed representation is, Gambit has an anchor sequence. So a sequence of 4 nucleotides. It looks for every k-mer of a certain length that starts with those 4 nucleotides and saves them. That's called the compressed representation. It has a database of all of the genomes from RefSeq, which is a giant curated database taken from NCBI. If you want to learn more about NCBI, please see one of our later courses. But the Gambit database has all of these genomes in it. Right now, it's a little bit over 4,000, but that database is constantly growing. We compare the compressed representation or the k-mer starting with that anchor sequence to the known sequences in the database. And from there, we can tell, based on the closest match of k-mers, what our sample or which species our sample belongs to. So you can see here that this is the smallest number. That's because this number is the Gambit distance. So we look at how distant our sample is from all of the samples in the genome, and based on the one that's closest, we know which species, or minimally usually the genus to which our sample belongs. This one is a very computationally efficient algorithm. It allows for rapid querying of many samples against a database. From there, once we have taxonomic identification, we can proceed with annotation. We use Proca and Bacta in our bacterial workflow at Theogen, but there are many different genome annotation tools available that are all, or many of them are open source on GitHub. This allows us to go gene by gene through the genome and assign names and functions to these genes. Predicting gene functions is a complex process. If you're interested in reading more on that, I would recommend starting with hidden Markov models. But suffice it to say for now that we know what certain genes look like, especially when it comes to AMR genes, and we can identify them in the genome using an annotation tool. Finally, we have genomic characterization. This is the process of looking at the markers in the genome, the sequences in the genome, and assigning phenotypic qualities such as antibiotic resistance. In addition to antibiotic resistance, which I've mentioned a number of times, we can also do taxon subtyping. So if you're looking for a serotype, a serovar, or some subtype that can tell you more about this pathogen and related pathogens, we can do that with bacterial characterization. And then plasmid detection is very important as well, because often AMR genes and other clinically relevant genes are found on plasmids, and these plasmids can be horizontally transferred between bacteria of different species. So even if two samples are of different species, they could share a genetically identical plasmid. Viral characterization often involves subtyping also, however, slightly different schema for the subtyping, where you'll get lineage and clade assignment. And then, of course, antiviral resistance mutation detection, such as Tamiflu resistance in influenza. So there's several different ways to perform genotyping, or sample characterization, if you will. Common genotyping methods are read mapping and gene alignment. These both sound like the same thing, but I assure you they're different, and I'll demonstrate that in a second. They both rely heavily on databases. So if you don't have a database of known genes or genomic features, it's going to be hard to employ either of these techniques. There are many different tools that do this. Often they are taxon specific. So Salmonella has very specific Salmonella genotyping tools. Concordant results from these different tools are very, they produce a large degree of confidence in bioinformatics scientists' minds when you have two different approaches that both arrive at the same result. So read mapping, what does that do? We are aligning our reads to a reference sequence. And so we take our read data over here coming out of our MySeq. This is paired end Illumina data, and we align it to our reference sequence. And this is a really good time to point out what this depth of coverage concept is. So you can see we have sequenced many parts of the genome multiple times here. You can see certain parts have greater depth of coverage than other parts of the genome, and we can be more confident in the sequence there. Now, if we look at read mapping, we're trying to genotype our sample. We're aligning our reads to the reference sequence. One of the most commonly utilized algorithms for read mapping is the BWA, Burrows-Wheeler alignment algorithm. Definitely read more into that. It's a super interesting algorithm. But essentially what we're looking at here, by mapping the reads to our reference genome, we are able to determine first the coverage of our each loci in the genome, the percent consensus. So how in agreement are the reads at every position across our genome? Is there anywhere where they are in disagreement? And what does that say about our sample versus the reference? So here we have an example of some non-matching reads, or reads that are not in consensus with one another. And we have what we would call 75% consensus that there is an A in this position, where in the reference genome, there is a C. So if there's an A in our sample and a C in the reference genome, that would be considered a single nucleotide polymorphism in our sample relative to the reference genome. And that single nucleotide polymorphism, it may do nothing. It may be a synonymous mutation, meaning that codon, that group of 3 nucleotides here, is still producing the same amino acid residue in the resulting protein. But sometimes these mutations can result in unique phenotypes, unique characteristics being conferred to the pathogen in question, such as antimicrobial resistance. So here we have 4x coverage with 75% consensus that there is, in fact, a single nucleotide polymorphism in this position. So read mapping, another way of using this is you have a database of reference gene sequences. So not genomes, but individual genes that we know encode certain proteins that have certain functions. So gene 1 here can be a gene that encodes a protein that breaks down a common antibiotic. Gene 2 here can be a gene that encodes a protein that is a toxin that kills people. And we can align our reads to these genes to see which of these genes are present in our samples. So we align our reads and we see, okay, we got a lot of reads aligning to gene 1 and gene 4, a couple to gene 2 and gene 3, but none to gene 5 and gene 6. So we can go ahead and say gene 1 and gene 4 are identified in our sample X. Now, gene alignment is slightly different. Here we have our sequence, and we assemble our genome assembly, and we align the reads to the assembly. And based on how well the genes align to our assembly and which versions of the genes align to our assembly, we can then make a sequence type prediction about our sample. So one thing to consider is that when we're doing gene alignment, the metrics might be a bit misleading. Percent identity and percent coverage are two of the metrics we use to assess how well genes aligned to our assembly. So here you have gene 1. It's got 99 or 90% identity and over 95% coverage. So 90% identity means 90% of the aligned sequence is matching, and it's covering 95% of the gene. Here we have 100% ID, 100% identity, which means there's no mismatching nucleotides, but we only cover 10% of the gene in our assembly. So which of these two matches is better? Is it better to have 100% identity, or to have 90% identity and only 95% or up to 95% coverage, over 95% coverage? We would say that it's better to have the 95% coverage with the 90% identity. That is more, that gives us a more confident call that this gene is in fact in our genome here. So in this case, we can safely say that gene 1 is in fact present. It might be a homologous allele. There might be a couple of mutations in our genome that has changed this gene ever so slightly, but it is in fact in our assembly there. So MLST kind of takes this idea of gene alignment and takes a database of many known gene alleles. So different versions of the genes, different mutants of the genes, still the same gene. So there's many different versions of gene 1, where it is very similar, but it can obtain some mutations along the way. So we take this database, we align our genome to this database of known gene types, and we can tell which allele is at each of the positions. So gene 1, if you imagine these in order in our genome, you can have different versions of the gene at each position. Based on the version, we assign a numerical code that is called the allelic profile or the sequence type. MLST, multi-locus sequence typing, is one of the most common sequence typing methodologies used in bioinformatics, and it facilitates conversations about pathogens that would otherwise be very difficult to have, saying, oh, I have this sample that has this version of this gene, this version of this gene, this version of... Instead, you could just say, we have sequence type 71. And everyone knows that sequence type 71 is the type that has a particular virulence factor or a particular phenotypic quality of interest to the public health or clinical microbiology community. So this allelic profile will be assigned a sequence type number. That sequence type number is how the conversations typically take place. CG MLST. So in regular MLST, you take a set of housekeeping genes and you look at those and you have a numerical identifier that is based on the allelic profile. In CG MLST, you take the entire core genome. And what the core genome is the set of genes that are found in every sample from that species. So the regular MLST, it's 7 genes. Core genome MLST, it's thousands. From core genome MLST, we're doing essentially the same process. We get a much longer set of alleles, but that is all boiled down to a sequence type. So again, you could have sequence type 71 in a core genome MLST, but the allelic profile will be 1,000 different allelic, the allelic code will be 1,000 digits long. So in summary, molecular biology facilitates all sorts of analyses in the laboratory. It allows us to characterize pathogens, to connect similarly related pathogens, and to learn more about novel pathogens. Next generation sequencing is one of the greatest inventions of our time. It has really allowed molecular biologists to learn so much more about pathogens and the genes inside of them and what they do, to learn about how disease propagate through a population. We have used next generation sequencing to get really high resolution transmission networks. Bioinformatics is all of the computational methods that we use to analyze next generation sequencing data. It allows us to characterize and track pathogens over time. And there are many freely available bioinformatics platforms out there. Some web-based platforms that I'm a fan of, Terra.bio, Galaxy, CLC Genomics, and Nextflow Tower. These are all openly available platforms. There's a ton of open source code on GitHub. If you're interested in getting started in bioinformatics, that's the first place to start, but also feel free to reach out to me. I'm super passionate about people learning bioinformatics and learning about genomes and the amazing technologies that we have to analyze them. So in conclusion, if you need any more support, please feel free to reach out to us at Theogen using our support email, support@terrapublichealth.zendesk.com, or you can reach out to me at Theogen, frank.ambrosio@theogen.com. Thank you all for watching this first session. There is a second session associated with this, so please check out that video as well. And good luck with all of your analyses. And I hope to see all of you in the next Bioinformatics conference. Take care, everyone.